Hello, everyone. Today I want to talk about the infant microbiome in sickness and in health. And this is part two of two parts, and you can find the first one here on the Layer Origin website. Let me give a brief review of part one so you know what we're talking about. This concerns a newborn who had a very traumatic birth. Hemoglobin is 3.7 at birth, 10 days in the neonatal intensive care unit. But the main thing I want to focus on is the fact that he was born by C-section and had antibiotics. And if you're born by C-section, you don't get the benefit of passing along the beneficial bacteria through the vaginal birth. And then if you get courses of antibiotics, he had two different antibiotics that will wipe out whatever good bacteria you do have, which almost certainly will lead to a leaky gut in newborns. And just by review, he had two major competitors occurring in his gut. First, you have Bacteroides fragilis, which is at least a pathobiont, if not pathogenic. And certainly when you're at about 57% here, it's highly, highly inflammatory. And his Bifidobacterium, this is his total Bifidobacterium levels, they start at 30%, went all the way down to 12%, and then rebounded. And they rebounded because his practitioner suggested that the mom give him Bifidobacterium longum subspecies infantis. And then at the strain level, you're talking specifically about strain EVC001. And that was good news to see the green crosses there, the green X's rather, are time when the B infantis was administered. It was only given to him for 2 weeks, and you see this dramatic, essentially a complete wipeout of the B fragilis, the inflammatory one, and then a nice rebound even beyond what he was, 33% of the Bifidobacterium. So that was a good result. But he was tested again after this last month in September, and some more just incredibly dramatic results here. So this chart is very different looking. The previous tests were done by Ombre and then uploaded to Biome site, which you get those really nice comparative charts. This chart I just made on my own because the September test was done by TinyHealth, and it's a completely different type of test. I'll talk about that in just a moment. But the result was just absolutely incredible. So you've got the Bacteroides fragilis, and then this is the total Bifidobacterium again, and it went from a low point, his Bifido, 12%, all the way to 81%. And I'll show you where the B infantis was administered, right around there. Again, only 2 weeks, and then took him off of it. Hasn't had any probiotics since then. He is being breastfed, so he's getting the human milk oligosaccharides through that, which feeds the Bifidobacteria. And you see that not only was his total Bifido at 81%, his total B longum infantis was at 75%. Now, I have a question mark there by EVC001. We're not certain that all 75% of that B infantis is this particular strain. At the time of testing, TinyHealth was not able to detect this. They can detect certain strains. They were not able to detect this particular strain, but they're changing the way they do their, I think it's their bioinformatics, and they believe that within a month or two, we will be able to go back and actually see if this was EVC001. But it makes absolute sense to me that if this is what is being colonized, and this is the only probiotic that he was given, that this is almost certainly the EVC001. And this is just a quick comparison here between the Ombre does the 16S sequencing and the TinyHealth does the shotgun sequencing. The shotgun is more expensive, sometimes twice as much or more, but also it can go down to that subspecies, even the strain level. And again, TinyHealths are improving on the strains that they can pick up on. So this was a perfect time for this baby to have a TinyHealth test done. This chart comparison shows the difference between Normal breastfed infants that have the control group on the left, they have up to 40% bifidobacteria. But the right, the EV, that should be EVC001, they went just skyrocketed up to about 80% and remained. So they were on this B infantis for 3 weeks total, but remained to at least 4 or 5 weeks after that. That's as far as this testing went. But if it's similar to the way this baby responded to EVC001, probably continued on as long as he was being breastfed and had the HMOs to feed the B infantis. For some of this video, I want to talk about a topic that might be new to some, the difference between secretors and non-secretors. Now, this is in regard to the ABO antigen, blood group antigen. So most of us know that your blood type is either A, AB, B, or O, and those are antigens in your blood. Well, these same ABO antigens for a secretor are secreted in other body fluids such as tears and saliva. And this is men and women, same can be either a secretor or non-secretor, and about 20% of the population are non-secretors. So what that means is they don't have an FUT gene, that's just that turns on the ability to secrete in those other body fluids. Also means that for breastfeeding women, they have no 2FL, that is the most common type of HMO in breast milk. There's 150 to 200 different HMOs. Some of them are just, almost infinitesimal. But 2FL is by far the most abundant type of HMO in breast milk. So that's not in breast milk or the gut mucosal lining. Non-secretors, and this would be men and women, have lower levels of bifidobacterium. The only benefit is that they're actually immune to rotavirus. So that's an extremely nice side benefit there. And there is a slight increase for some disease. Secretors do have the FUT gene. The nursing moms would have 2FL in the breast milk, and as a result, they have higher levels of bifidobacterium, but they're not immune to the rotavirus. So this chart is really helpful in regard to this particular baby. You see on the left there, his bifidobacterium levels, again, all the way down to 12%. And on the right, you've got 4 bar graphs. The cesarean minus is a cesarean-born newborn that is a non-secretor. So the minus represents the secretor status. It's a non-secretor. And then to the right, that cesarean-born secretor. Then you have vaginally born non-secretor and vaginally born secretor. It's interesting because the secretor status for the vaginally born infants is almost makes no difference whatsoever. So that's good news. But cesarean, you see a huge difference between secretor and non-secretor. And that, again, is related to the types and amount of HMO in mom's breast milk, because not only non-secretors don't have the most abundant type, 2FL, their overall total amounts of HMOs in the breast milk are lower. And our best guess is that this child's mother is a non-secretor. Two reasons why. One, just looking at his extremely low levels of bifido, the 12%, you see the cesarean-born non-secretor at about 18, 19%. So his are even lower than that. Also, the mom does not recall ever having had a rotavirus. So even though she did not have a genetic test, that's our best guess. And at least initially for this newborn, all those combinations, that would just, one more small strike against him in his early life. But the main issues were, again, that he was born, not so much the secretor status, but he was born by cesarean and that he was given antibiotics. This shows the most abundant type of HMOs after 90 days of lactation. You'll see that over the lactation period, the total HMOs will decrease in total amounts. And I'm also putting up there in the yellow, the non-fucosylated HMOs. Again, these are just different types and different chemical structures. They're fucosylated, non-fucosylated. That's going to come into play a little bit later in the video here. But quoting some research studies, it's the same research study as the previous slide. In infants of non-secretor mothers, there seems to be a trend towards increased abundance of enterobacteria. Those are the gram-negative inflammatory type bacteria. They tend to be inflammatory, which we also observed in the cesarean-born infants. Many members of the enterobacteria are inflammatory, some even pathogenic. Fucosylated oligosaccharides are decoy receptors for many pathogenic bacteria, including members of the enterobacteria. Hence, they have a potential to reduce their adhesion to the gut, thus protecting the infant. So again, that's the benefit of the fucosylated type. In addition, fucosylated HMOs bind enterotoxin produced by E. coli, inhibit its effects on intestinal cells, and reduced LPS lipopolysaccharide. That's a gram-negative produced inflammatory molecule. Inflammation in gut epithelium. Fucosylated HMOs may thus reduce the colonization ability of enterobacteria, as well as their inflammatory effects in the infant gut, particularly among prematurely born infants. So you see just multiple benefits of the particular type of HMOs, these fucosylated HMOs. Here's the composition. The solid portion of breast milk, if you take out the water, number one would be lactose, number two would be lipids or fats. Third most abundant solid in breast milk is HMO. And you see there the range is between 5 and 15 grams per liter. And you'll see that those decrease over time. I'll show you that in a moment. So that's just your typical composition of breast milk. Now, here's the difference between a secretor and a non-secretor. As I said, a secretor has 2FL, the most abundant type, but the non-secretor has 0 2FL. Now, they compensate for that by increasing especially the LNT, LNFP2, and the LLNT. So they do get some compensation there. But again, the total amount of HMOs in breast milk is also going to be decreased. Here is a chart that shows the HMO concentrations throughout the period of lactation. And you see there in the bottom row, it goes from 9.4 to 6.2 over the 90 days. And you would assume that it just keeps decreasing very slowly. So that's interesting that the infants benefit from a lot more HMO in the early days, which just makes sense. Another research article, abundant HMOs in human milk, such as 2FL, 3FL, and 6SL, were below detection limits in the fecal samples from infants in the EVC001 group. Let me make sure we understand what's going on here. So these infants, they're being breastfed and also receiving this probiotic EVC001. So they're measuring the amount of HMOs in fecal samples, and they're saying that it's below detection limits. And that is because the EVC001, again, that's the strain of the B. infantis, is so good at consuming HMOs in the breast milk that they literally cannot detect any HMO in the fecal sample. Going on. Importantly, the abundance of HMOs in the fecal samples were negatively correlated to the absolute abundance of B. infantis, which makes sense. The more B. infantis there is, the less HMO there's going to be. Taken together, B. infantis and the EBC001 group provided the microbiota the capacity to metabolize HMOs, which resulted in better metabolism of these molecules in the preterm infant gut. These results are relevant, as recent data suggest. Metabolites found in fecal samples of term infants exclusively breastfed and colonized with EBC001 can directly influence the development of the immune system. So that is to say, when you have lower levels of inflammatory bacteria and higher levels of the Bifidobacterium are essentially anti-inflammatory molecules produced by those bacteria, it is better, much, much better on the immune system for newborns, but also it affects later in life as well. Another paragraph from that same research article. Therefore, the results generated from this study and from previous studies utilizing EVC001 support that this strain provides infants with the capacity to metabolize HMOs with positive results in immune development, as we've been saying, as well as decreasing the risk of developing comorbidities common to prematurity and related to enteric inflammatory conditions such as necrotizing enterocolitis. While it is widely accepted that preterm infants benefit from human milk, here we demonstrate the value of matching the right bacteria with diet, and referring diet now to probiotics from the EVC 001. This symbiotic approach to care in the NICU has important implications for stabilizing the gut by fostering the establishment of an infant appropriate gut microbiota to maximize nutrient acquisition from human milk. That is to say, you know, preterm infants spend a month, sometimes 2 months or more in the NICU. And this whole time, if they're not receiving HMOs and or the probiotic B. infantis, they're going to suffer a lot more immune issues than if they would receive both of those while still in the NICU. Another research article saying very similar things. Immune microbe interactions early in life influence the risk of allergies, asthma, and other inflammatory diseases. Breastfeeding guides healthier immune microbe relationships by providing nutrients to specialized microbes that in turn benefit the host's immune system. Here we show that a lack of bifidobacteria, in particular depletion of genes required for human milk oligosaccharide utilization from the metagenome, and uh, Bifidobacteria consumes HMO, but different Bifidobacteria consume some HMO or a lot of HMO, as the B. infantis does. So they're saying the better that the Bifidobacteria can consume HMO, the better end result. And the result is associated with systemic inflammation and immune dysregulation early in life if you have that depletion. But in breastfed infants, given B. infantis, this particular strain, which expresses all HMO utilization genes, intestinal T helper 2 Th17 cytokines were silenced and interferon B was induced. In other words, there is anti-inflammatory reduction of pro-inflammatory molecules and increase of anti-inflammatory molecules. One more research now, we're switching gears a little bit. We're talking about the pathogen, Bacteroides fragilis, this particular strain, shows the highest HMOs consumption rates from all bacteria analyzed, ranging from 25 to 90%, nearly depleting all structures from the HMO pool with high degree of polymerization. Now, let me stop there and remind you that we're talking about not about B. infantis, but the good bacteria, but B. fragilis. And what this is saying already, that B. fragilis loves HMO and is consuming it, again, almost depleting all of it. So we're going to get this interesting competition going here between Bifidobacteria and the B. fragilis. Unlike Bifidobacterium infantis, B. fragilis exhibited a glycan utilization pattern where the relative utilization of non-fucosylated glycans was higher than the consumption of fucosylated oligosaccharides. So just saying that they like all HMOs, but they tend to prefer the non-fucosylated HMOs. Now, this is an absolute fascinating chart. I call this the colonic battleground. You've got B. infantis, Bacteroides fragilis, Bacteroides vulgatus, but focus on the B. fragilis, which is in the light gray, and the Bifidobacterium infantis. Now, this is B. infantis, but it's a slightly different strain. It's not the EVC001. You see in almost every single bar graph that the B. fragilis is out consuming the HMOs as compared to the Bifidobacterium. So we're talking about this competition to consume the HMOs, but the difference is, now you would say, if the HMO is feeding the pathogenic bacteria and they like it just as much as the Bifidobacteria, are we not harming a child by giving them HMO because it's feeding the bad guys as well as the good guys? Well, that would be the case if this was in a laboratory setting. Now, this is a laboratory test here, results. And they're saying if you incubate them separately, this is what's happened. This is what happens. But the gut is just a battleground. That's why I call it a colonic battleground between the good guys and the bad guys. They're constantly fighting for dominance, and we want the good guys to win. And here is a unique benefit that Bifidobacteria have over B. fragilis. However, B. infantis is the only known organism capable of consuming all HMO structures in mother's milk. That's literally the case. As far as we know, it consumes every type of HMO due to its unique genetic architecture. Specifically, B. infantis contains clusters of unique genes that encode for proteins required for the capture, uptake, deconstruction, and metabolism of HMO in human milk. Consequently, we and others posit that human milk is a selective growth for B. infantis, the engraftment of which establishes colonization resistance in the infant gut by lowering the pH. That is such an important phrase at the end of that paragraph. What it's saying is that B. infantis, because it produces lactic acid, uh, that is fine for the bifido. They can tolerate a lower pH, but the Bacteroides fragilis does not tolerate that lower pH, and it begins to kill the B. fragilis. So that's the competitive advantage. Even though they're both competing equally for the HMO, the lowering of the pH due to that lactic acid is what gives that competitive advantage, and significantly so. So much so that in this case of this infant, it virtually eliminated the pathogenic bacteria and sent the bifido all the way at 81%. One more, bring a different idea here. In fact, the apparent lack of major functional differences between EVC 001 and the ATCC 15697, that's just a different strain of B. infantis, suggests that these strains share many phenotypic and health promoting features related to the ability to metabolize HMOs and colonize the infant gut. I just put this slide up here to let you know that the EVC 001 isn't the only strain of B. infantis that does pretty much the same thing. These two strains are almost identical. Last research here. This is showing the consumption of various types of HMOs between secretors and non-secretors. Secretors obviously, again, have the 2FL, but it's interesting that the LNT and the LLNT and the non-secretors, the EVC 001, which pointed at by the green arrows here, greatly, strongly prefers the type of HMO produced by non-secretors. So that is a little competitive advantage and sort of makes up for some of the decreases in non-secretor mothers. So by way of summary, using the EVC 001, total bifidobacteria in this infant went from 12% to 81%, and 75% of that was the B. infantis. Using that same probiotic, total Bacteroides fragilis went from 55% to 1%, virtually wiped it out. In general, C-section babies have less bifidobacteria, and if they have non-secreted mothers, they have even less bifido, which is why, in this case, the practitioner suggested that they take the probiotic. B. fragilis heavily competes with Bifidobacterium for utilization of HMO and prefers HMOs that are more common in non-secretors, the non-fucosylated. B. infantis, EVC 001, consumes all types of HMO and prefers HMOs more abundant in non-secretors. B. infantis produces lactic acid, which lowers colonic pH, which in turn inhibits pathogens such as B. fragilis. And finally, we've seen that 2FL, which is absent in non-secretors, helps fight pathogens and reduce inflammation. So that's an added benefit. So I hope this was interesting, helpful for you, and I appreciate you watching.